Hi all, and welcome to today's webinar, Inside the Target ALS Annual Meeting, Research Updates for Our Community. I'm Stephanie Ishu, Director of Marketing and Communications at Target ALS. Before we begin, I just want to share a quick background about the organization. So Target ALS is a nonprofit that's breaking down barriers to ALS research so that we can find effective treatments. And this is all in service of our ultimate vision of building a world where everyone with ALS lives. We carry out this mission through our unique fund, enable, conduct model in which we fund really compelling ALS research. We enable ALS researchers across the globe to perform their work by providing a suite of critical tools and resources like data and samples. And we conduct our own ALS research through our ALS Global Research Initiative, or AGRI, that will help us answer longstanding questions that the field has about this disease. This all converges at our annual meeting, happens every May, and that's what we're here to talk about today. And I'm joined by an awesome group of panelists here, Amy Easton, Christopher Donnelly, and Lauren Mifflin. And I'm going to give each of our presenters a moment to introduce themselves, and let's start with you, Amy.
Hi, everyone. Welcome to the webinar today. I'm Amy Easton, VP of Scientific Programs for Target ALS. I'm a neuroscientist by training and have dedicated more than 20 years of my career to discovering new aspects of disease biology and new therapeutics for neurodegenerative disease, first in biotech and now for the foundation. Happy to be here today with you all.
Thanks, Amy. Chris, how about you?
Hey, thank you for inviting me to talk. My name is Chris Donnelly. I'm currently an associate professor and section chief of neuromuscular research at UT Southwestern. Prior to that, I was at University of Pittsburgh for about 10 years and was a scientific director of the Live Like Blue Center for ALS research there. My lab studies ALS and mainly focusing on TDP biology. And I've been, talk about this, but I've been to Target ALS meetings and a lot of capacities, including as a trainee investigator and funded investigator, as well as just an observer.
Thanks, Chris. And Lauren.
Hi, thanks so much for having me. My name is Lauren Mifflin. I'm a principal of company creation at Frazier Life Sciences, a biotech venture fund. In that capacity, I spend a lot of time building early stage companies across therapeutic areas and modalities. But by background, ALS is near and dear to my heart. I started working on ALS research because my aunt passed away in 2015. So it's something I've seen as a family member of someone suffering from ALS. In addition, I did my graduate work on the RIP kinase program that was eventually moved into phase 2 clinical trials for ALS. And so I've spent a lot of my time thinking about neuroinflammatory pathology in ALS and other neurodegenerative diseases.
Thanks, Lauren, and thank you all for sharing. So before we dive into the updates from the meeting, we wanted to share a little bit about the meeting itself. So Amy, could you tell us what the annual meeting is and why it's such an important program for Target ALS?
Sure. Thanks, Stephanie. So first of all, the annual meeting is held in Boston every year, and it's an opportunity for our grantees to present their most recent data, both advances and challenges in ALS research. And they're presenting this to each other as well as to a really wonderful cross-section of constituents, from venture capitalists like Lauren to academics like Chris to other nonprofits and patients, as well as biotech pharma. So it's really bringing together top scientists from around the world to talk about their research, studying basic mechanisms of disease biology or basic biology, and studying development of biomarkers that are so critical for diagnosing patients and getting them to the right clinical trial, and developing new ideas and new therapeutics and moving them forward towards the clinic. And what's so interesting about Target ALS is that we fund multidisciplinary consortia. We bring them together with the goal of breaking down those silos that traditionally have kept people in academia and biotech and other areas from working together. So we fund those multidisciplinary teams to talk about their work. And it's very exciting to see all the advances that have come together in this particular meeting this year. I think this year's meeting really embodied our ideas around creating and nurturing an innovation ecosystem, where we had an amazing contribution from all constituents present at this particular meeting.
Thanks so much, Amy. And Chris, you've attended several Target ALS meetings, as you've alluded to throughout your academic career. So could you tell us a little bit how the meeting has evolved from your first time coming to now?
Yeah, so I attended, I guess, what was the first Target ALS meeting in 2013, and there were some versions of it before that as well when I was a postdoc at Johns Hopkins with the great Jeff Rothstein. And what I can tell you is that during those early years, it was very academically focused, where you mostly had a much smaller group of researchers and clinicians. And this was sort of just pre-Ice Bucket Challenge, I believe. So the interest in ALS wasn't nearly as robust as it is today. And so what I've seen over the years from my experience is a real momentum of having industry partners and venture groups interested in kind of getting into the ALS space and developing therapies from like an early stage um, level as someone who has done that or tried to do that ourselves, uh, where we went from just having a few to having really a large number of companies, big and small, and a large number of startups kind of not afraid to start studying ALS, and also working with the investigators at the meeting. In the beginning, again, it was a smaller group, but what it has allowed us to do is sort of, the beginning was sort of more focused on what genes we can discover, why do motor neurons die, um, but now we're kind of moving to a stage in the field which was unheard of at the time, where we're actually taking these basic science questions and working with partners to actually develop therapies for those mechanisms that we talked about years ago. And that's pretty exciting because it's that was completely alien at the time. Um, and so I think beyond that, we're also talking about things that we as basic scientists don't think about as much, such as how to identify the right patients for clinical trials. What does a good clinical trial look like? What does a successful drug look like? We kind of have a pipeline. We kind of know now from tofersen and the SOD1 ASO. So I think sort of the meeting itself has evolved where not only do we see more interest from industry, pharma partners, and startups, but they're not afraid to get involved, which is something that they were, I think. And it also kind of gives a critical opportunity for us as academics who maybe have an idea that we think, hey, we see this and we think that this is something we can do, but we don't know how to take it to the next stage. This is not what we're trained to do. We have an idea. How do we make it to get it to a translate? And the meeting really provides a nice format for that, and you're seeing it really quite often as we've seen with several of the presentations this past year.
Thanks so much, Chris. And Lauren, from your perspective, as someone coming from venture capital, why is this an important meeting to attend? There are so many scientific conferences. So what sets Target ALS's annual meeting apart?
Yeah, I mean, I think Amy and Chris both touched on this, but the fact that the consortia that are funded are inherently interdisciplinary, I think brings the best of every world to every project, right? You get industry level drug discovery tools and or resources applied to some of these consortia. You get their venture investors and their boards providing input onto what a fundable data package looks like or something pharma would be excited about. You have cutting edge academic innovation and insights around, what biomarkers or targets we might be able to go after. And every project kind of folds all of those together. So I think the way that Target ALS structures these grants and programs requires people to bring the best of themselves to each of these programs. And so it's really fun to watch things move forward, frankly, I think faster and better than they would have otherwise. And so, a lot of the ones that even in the, I think, 7 years I've been coming to this, seeing some of the ASO programs go from we have a hit on a slide to someone can make an antisense in 2 years to now we have patient data coming out. That's pretty wild. And I think part of the excitement from the industry side of coming, one, Target ALS has a pretty awesome scientific advisory board reviewing these grants. It's a lot of folks I've had the pleasure to work with in industry, and they're really committed to the annual meeting. They're often the moderators of sessions for those of you who've only joined online. But in their day job, they're highly credible and impressive drug developers in their own right, who are committed to making sure that the right grants and consortia are getting funded. And I do think another part that's unique here is because it's not first and foremost an academic conference, there's less of an impact of, in my opinion, what is going to get published first and foremost, and what is going to get federal grant dollars first and foremost. And so I find the program's always a really great mix of, the biggest, most important topics, right? So things like TDP 43 that are getting a lot of attention and insights right now, but always a few other sort of counterculture topics, whether they're a hit we don't understand from GWAS, whether there's something that might be an interesting biomarker or something that came out of a small molecule screen that we're working to understand. There's always enough different perspectives that, I think it helps people think not just about what do we know about the disease now, but where we could be going in the future, depending on how a few things play out.
Yeah, thank you for sharing that. And before we dive into the science that was presented this year, it was quite an exciting conference. Just want to give Amy or Chris an opportunity if there's anything you want to add on the uniqueness of the meeting before we move on.
I think we already touched on it. I think the most important part is that this is an international global meeting. This is a rare disease, so we really can't afford to have individuals working alone, going slowly. We have to work together. And I think, again, this year's meeting really allowed for a lot of international collaborations to exist. The researchers themselves love this part of our program, but most importantly, it's benefiting patients by advancing the science faster.
Yeah, and I'll just add the one thing that I really enjoy about the meeting every year is the Springboard Fellow talks. So the Springboard Fellows are the sort of late stage postdocs who are transitioning from, trainee to trying to become an independent investigator with an ALS research program. I was very fortunate to be one of the first to get those back in the day. And it really helps you secure a position if you want to stay in the field. It's not easy to stay in research, let alone in the ALS space. It's again, it's some really good people in it. It's hard to find positions, and but the quality of the trainees, and the kind of creative topics and approaches that they take are always something that none of the more established or people who've been doing it for a while have. They don't you won't see that from the more established labs generally. So they always take sort of little higher risk, really interesting projects with a very unique approach. And every year they get more and more impressive. It's actually kind of intimidating from my perspective, but I think that is really one of my favorite highlights of the meeting. And it's not something that you see where you have trainees giving an opportunity to speak in front of such a large and international audience at many meetings. That isn't a common thing.
Thanks. Yeah, we're very strict on our grant programs. They're either collaborative or they're bringing in this next generation of researchers so we can get the best in ALS research. And so now let's dive into the science. As Amy had stated earlier, we have these three major focus areas: basic biology, biomarkers, and drug discovery. So we'll go through each of them and we'll start with where it all begins, understanding the disease itself with the basic biology. So Chris, what is the current expected dogma of sporadic ALS, and what did you see from this year's annual meeting that maybe changes our view?
Yeah, that's a really good question. So I think the dogma of ALS has changed over the years in different ways where we focus on different things depending on if a mutation is discovered and whatnot. I think right now where everyone is kind of focusing on their attention to is the pathology that every patient has. And it's called TDP proteinopathy or TDP pathology. And what this is a RNA binding protein, TDP43. It lives in the nucleus generally. Um, and in patients, nearly all patients, like 97% of ALS patients, and also other diseases like FTD, about half of those cases, and even some Alzheimer's cases, a large subset of them actually. You see that this protein is no longer where it should normally be in the nucleus, where the DNA is, and this other molecule called the RNA, and it's mislocalized to the cytoplasm and often forms like these clumps or aggregates. And for years, we've known that that occurs. It was identified as TDP being the primary component in, I believe, 2006 by Virginia Lee and John Janowski. But we never really knew the consequence of it or why it was a problem. And so it's kind of an evasive and difficult protein to study because it's so important for normal cell function. And if you mess with it in any way, it seems to be toxic to most cells. And so the things that we've been seeing that are, I think, very exciting is that in the past couple years, and particularly this year, we're starting to see that we can identify the consequences of this protein not working, and we're starting to get better models of how to kind of model this event in a dish because it's not something we can very easily model. So I think the modeling is getting better, which has historically been very challenging. And we're also now being able to see the consequences. And when you can combine that with what these large data sets that Target ALS and many of the investigators who were part of it have developed, we can kind of compare and say, this is what happens when this protein is not functioning in our cell model or in our animal model, whatever. But then is that important? And do we see that in patient data sets? And so that's kind of been this really nice sort of link between the two. And it's allowed us to one, validate whether or not our models are really even answering or addressing the right thing, which has been historically a major challenge in our field, is how do we model this disease? And we can now compare it to patient tissue and use, or patient samples, and use sort of our understanding of what's actually going on there. But then the really exciting part, I think, that we've been seeing now is now that we know some of the consequences of this happening, when TDP is not working, um, can we target some of those events? And so there are two presentations that I'm sure we'll hear a little bit more about, but where we're actually targeting some of the downstream consequences of TDP not working well, not properly functioning and doing its job. And there are now clinical trials targeting those. So while we still, so now we're going to have an answer as to whether or not is that sufficient, targeting some of those downstream events, is that sufficient to kind of give a therapeutic benefit for patients? And that is really fascinating because I mean, if you'd said that a few years ago, not many people would believe we would that quickly go from identifying this to actually getting this into a clinical trial. And that's what we want is right, fast progression from basic science to translation. So
Thanks, Chris. Lauren, do you have anything to add on that?
No, I mean, I think we'll just come back to this, but one of the reasons it's so important to have things like the Target ALS annual meeting is that, as Chris has alluded to several times, like genes like TDP43 that we have no question are involved in ALS pathogenesis and very important to neuro and neuroaging health in general are not easy drug targets. We're not talking about something like a kinase in oncology where you can find a pill and inhibit it and dose that, and you know you're going to stop what's happening downstream. These are really complex and multifactorial things. And so, even having one annual meeting to the next, looking at the nuances in our understanding of what part of the perturbation of TDP43 is driving which of the downstream pathology has been evolving really quickly. And so I think having many different groups with orthogonal interest motivations and tool sets kind of working on this has helped us move more quickly as a field here. But I think, there's still, as Chris mentioned, some ability to go downstream of TDP43 and fix one or two of the things that are perturbed. But how do we fix the whole pathway? And do we understand what fixing the whole pathway looks like? I think is still something we're working towards.
Thank you. And Amy, from the Target ALS perspective, first, anything to add? And then also, how does this affect how we set out for our next, let's say, round of grants or the areas that we're going into?
Yeah, absolutely. So absolutely agree with Chris and Lauren. They really spoke to different pieces of that beautifully. I would just add on from my own experience in drug discovery, the central dogma of where TDP43 is becoming mislocalized and aggregated and is potentially an important key upstream factor that leads to motor neuron degeneration. That dogma really evolved over time even after the time that I started doing drug discovery in 2005. So it's taken some time for us to put all the pieces of the puzzle together to kind of map out what is that pathway, what are the different pieces of that pathway, starting with the TDP43 itself. And that has brought, with the help of Target ALS funds and the research we enable and we conduct, a number of different companies that are really just at that moment of testing central dogma, and they're testing it in patients. They've accumulated a lot of wonderful data behind their molecules to target TDP43 itself. So I think I think that's going to tell us more about, is that the right thing to target? And then we have, as Chris was mentioning, a number of groups who are targeting the next events that are happening downstream, which is this change in RNAs that are normally tightly regulated in the nucleus. Now you're developing a number of aberrantly forming RNAs that we are calling cryptics. And both Curalis and Trace have molecules that are in the clinic testing those hypotheses. And we're going to come to this more, I think, in a little bit, but Curalis's clinical data are encouraging to suggest that you don't necessarily need to only target TDP-43, but you may have benefit even if you are targeting the next steps in that sequence of events. Second point that I really want to mention that came out very clearly across all of our basic biology researchers was a better understanding of the role of glia. So in the disease biology. So we know, of course, motor neurons are what's dying, but there are many different cells in the brain and spinal cord that contribute to maintain proper neuronal function. That includes oligodendrocytes that are myelinating those axons to allow them to fire and transmit messages one to the other, as well as microglia, the innate immune cells of the brain that are looking to protect the brain from viruses and other invasive substances. They're there to gobble up these aberrant proteins like TDP-43. And so altogether, that whole area, I would say, we've known for some time that there's some involvement, but in this particular meeting, there was a lot more data coming through to show that there's actual relationships between specific proteins and cell types and disease progression. So I think those are the kinds of events that lead Target ALS to help develop new funding calls to further develop those wonderful ideas that are coming from those basic biology projects now.
Thank you all. And so let's move into our next section, biomarkers. With this new and evolving context from our basic biology, what biomarkers are emerging and what are the challenges or exciting breakthroughs that are happening in this space? And Lauren, I'm going to start with you.
Great. As I think about biomarkers, there's a lot of ways to split them apart, but two macro categories that I'll start with is the notion of what biomarkers measure disease progression, what biomarkers, can we use to get a sense of how quickly someone's disease is moving, and if it's slowing at all ahead of actually being able to see changes in the ALS functional rating scale. And then there's the other biomarkers, which I'll think of as the biomarkers of target engagement. Do we have confidence that our drug is getting where it needs to get in the brain and the spinal cord, and that it's actually engaging its target in a robust way? And I would say we've made a ton of progress in both of these areas. So if we start talking about disease progression, likely most folks in the ALS community are quite familiar with NFL or neurofilament light chain. This is a protein that in cerebrospinal fluid elevates very quickly when neurons are damaged, unhappy in any way, shape, or form. And that can be because you've put a toxic drug into the CNS and you actually see rapid elevations in neurofilament light chain, or it can be associated with progression of a neurodegenerative disease. And from where I sit, which is often thinking about not just ALS, but other neurological diseases, I actually think ALS has one of the clearer and stronger stories about how neurofilament progresses and elevates as diseases continue. Why that's so important is that part of why there is an influx of biopharma and venture capital interest to ALS is that as you start to run early clinical trials, because the disease progresses quickly, much sooner than something like Alzheimer's if your drug is working or not. You don't need to wait for, 5+ years of someone's memory declining. You can see the changes in, bulbar or skeletal muscle control sort of in short time periods. The neurofilament time changes also seem to reflect that in ALS. And so it's something that as, folks are running clinical trials, they can serially sample in cerebrospinal fluid and really see if you're having an impact on that neuronal damage or degeneration over time. So I think that's one big category. And then on the target engagement front, does your drug work and is it getting to the cells where it needs to work? I think we've made a lot of progress on what I'll call the heavy hitter targets that are really difficult to measure. So TDP-43 being so complex, or something like C9 or F72, the biomarkers of what you measure to know if your drug is engaging with those targets in the right way are also very complex. And so there's been a lot of advances in things like, for example, antibodies to detect cryptic exons, which end up expressed downstream of TDP-43 mediated missplicing. So you end up being able to get to, because of the scientific advances in basic biology, better detection methods to tell if the drugs are going after some of these more complex targets that we're now starting to have therapies about. And so we need both to run the right clinical trials and both to write, a thesis behind backing an ALS company. And so it's encouraging that both have moved forward.
Yeah, it's very exciting to see from year to year how these nuances are helping change our understanding. Chris, do you have anything to add about biomarker development, challenges, and breakthroughs?
Yeah, no, I think what's also kind of exciting, even from a basic science standpoint, is to some of the biomarkers that Lauren had mentioned. We can model those, in a lab setting. And so now we can use that to, if we have an idea of a target or a pathway we might be able to engage to try to, if we think it might be important in disease progression or may eventually be a therapeutic target, we can use those both sort of in our lab as a reference point. And when we do go talk to industry or other groups to say, why don't we try this? We now have that as well. So it really kind of nicely brings things together. I also think the biomarkers as a whole, as kind of mentioned, what like this combination of them um, kind of tells us also or allows us to kind of understand maybe what the drug or therapeutic approach is actually doing, because maybe you're rescuing one thing but not rescuing the other. And so we can kind of start dissecting out what mechanism is the drug working on or the therapeutic approach working on in the disease. And, maybe one's going to be more important than the other. We don't know. And I think the other thing I would just say is that what I saw this past year that was kind of interesting as well, that beyond just sort of TDP biomarkers and others, we also are seeing some like inflammatory signatures and things that may also be used potentially as biomarkers down the line to better understand maybe further downstream consequences that we could potentially add that into sort of this larger panel of things to understand how what's going on in the disease as well as whether or not a therapy is working.
Thanks, Chris. And Amy, how is all of this informing our biomarker strategy moving forward?
Well, so yes, just to also add on that part of our strategy over the past couple of years has to really try to put as much resources as we can onto a number of different potential biomarker candidates and biomarker assays. And in particular, what you saw at this year's annual meeting were a number of different consortia taking different approaches to try to study what I mentioned earlier, the presence of these cryptic exons or cryptic peptides that might be floating around in the cerebral spinal fluid or in the blood of patients with ALS. And the reason that they're doing that is because it's so difficult to see differences in TDP-43 protein itself. That historically has not allowed us to differentiate ALS from healthy individuals or any other disease area. So again, the upside of having a number of different folks try different approaches is that they get to compare notes and they're sharing their research, both their advances and their challenges, together in the same forum. And I really love this. It's really important for everyone that they're doing this because it allows you to discard ideas that aren't working more quickly and pull together your resources to focus on the things that are working. So in that space, what I saw emerging are using highly sensitive assay platforms like the new ELISA to measure low levels of cryptic peptides in CSF. There was some breakthrough in that space, as well as breakthroughs in measuring cryptic exon mRNAs in extracellular vesicles that are secreted from the brain and can be found floating around in CSF and plasma. So I think, again, there were small steps in the right direction. A lot of difficulties doing that because these things are found at such low levels. And it's also, there are hundreds of them that you could be looking for. So you have to determine which ones could serve as the best biomarker and filter through all those. So I again think there were some advances there and looking forward to seeing the next step up in those advancements that won't just be around cryptics. It will also be more broadly around synaptic biomarkers, other kinds of biomarkers that are complementary to neurofilament light, which does tell you about damage to the neurons itself.
Thanks. Yeah, it's very exciting to see how all this is moving forward. And that brings us to our last category of drug discovery. So, Lauren, I'm going to start with you here, thinking with your VC background. What's exciting in ALS drug discovery, and how has Target ALS helped seed this pipeline?
Yeah, I think as I alluded to earlier, the types of targets and biology we need to go after here aren't always amenable to something like a simple small molecule. And that means that we need to be taking on these more complex ways of modulating targets. And that has historically had a big backlog of what I'll call delivery and technical risk, right? Whether that's AAV therapies injected into the brain, antisense oligonucleotides administered intrathecally. I think the good news is we're at a really great moment in time where those two things have kind of caught up to each other. We have a better understanding of TDP-43, glial, all of these different biologies and how they matter in ALS, and also which cell types they matter in ALS, as well as sufficiently mature platforms for either delivering, viral or lipid nonparticles, so non-viral, or oligonucleotide siRNA, that type of thing into the CNS. And so with the confluence of those two things, one of the really encouraging parts of this year is for two of three of those targets, we've started to have early clinical data come out. So as Amy mentioned, Curalis's program going after Stathmin-2, which is sort of downstream of the TDP-43 mediated missplicing in the cytoplasm. Trace's program going after UNC13A. I believe, Curistem and a few other groups are working on these targets. I think we have growing confidence that we're putting the right antisense sequence delivered in the right way to cover the target and be able to figure out if going downstream of TDP-43 is able to slow and ameliorate disease. So for me, it's just really encouraging because these are targets I first heard about at this meeting 5 or 6 years ago. And now we're getting to see the first, directionally positive patient data to suggest that we're on an interesting track here. There are a handful of other groups trying to work on ways to directly modulate TDP-43 in the CNS, which is a challenging undertaking. But again, I think we start to have some of the viral and non-viral delivery tools to do that. And maybe just tying it to the biomarker piece, I think it's really important that this is coming at a time where we can use CSF or brain imaging to be able to check, is it getting to the right cells in the brain? And, does that matter? Because I think, again, you need the confluence of those things, the right biology, the right way to deliver it, and the right way to measure it to when you get your phase 1 data coming out, be able to say, did this trial work? For which patients did it work? And for some that it didn't work, do we understand why? Is it that we didn't get enough drug to the neurons where we need it, or that we might not actually have the right target? And being able to deconvolute those things, I think, is really important.
Yeah, and of course, looking to our community, we can never move fast enough, right? But it is nice to see that, with that Curalis presentation and that announcement during the meeting was just very exciting to see that there's good things happening within these clinical trials for people living with ALS, and hopefully that continues. And Chris, anything to add?
Yeah, I mean, I think the two things I would add are one, just the rationale of what we're targeting. The hits that are being developed and or therapies that are being developed are not just, I mean, they're targeting real aspects of the disease, not just something where it's like, well, this is, affecting inflammation, so let's just see if we can try that. And I'm not saying that that's wrong, but it's like we know that these events that are being targeted are very specific, they're very centered around the disease. We know it happens in patients, and that alone is really exciting. It's also going to tell us that this approach is working probably more globally, or maybe we do need to be taking a bigger sort of just try anything approach. The other thing I would say, kind of going back to cure ALS and trace, the speed at which the discovery was made to which they're getting into patients is really incredible. I mean, I don't know exactly the number of years from the initial publications of these targets, but it's only a few years. And like that's what we want, right? We want to be able to move as quickly as possible and know. From a research standpoint, we don't want to spend our time doing something that's not working. It's a waste of resources, it's a waste of ideas. We want to know if it's going to work. And I think those are examples, along with others, beyond just those two, but of approaches that have moved so quickly. It's almost helping us develop, I think in partnership with industry, venture, et cetera, develop sort of a pipeline to move as quick as possible. And a lot of that is probably based upon what we know about the SOD1 ASO as well. So I think that's a big, 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 big step that we didn't have, 6 years ago, 7 years ago. So.
Thanks, Chris. And Amy?
Well, it's hard to pick which project is most exciting. There's so many exciting ones, but one thing I really wanted to point out from this particular meeting is the work that was being shared on patients who carry a C9orf72 repeat expansion, and what we're doing for that patient population. So, first of all, we know that there has been a trial that was sponsored by Biogen, partnered with Ionis to develop antisense oligonucleotides to target the sense strand. And there's been a lot of postmortem work, if you will, on the data that came out from that trial, including Biogen's own research that was presented at our annual meeting. And so I think the postmortem data show that there's actually a lot of good reason to target the antisense RNA strands. They're more abundant, they're correlating with TDP-43 pathology in the brain and spinal cord. And so they may be a more viable target, or at minimum, you may target both of those strands together and not just purely the sense strand. So what came out of the consortium that consists of Ionis Biogen and Johns Hopkins is more information from their preclinical work to support advancing ASOs to target that antisense strand, and also including differentiation and potential explanations for why that sense ASO may not have worked initially and why targeting the antisense ASO may be an effective therapeutic going forward. So there was really a lot of nice cell-based work and animal model work supporting the advancement of that idea towards the clinic. And also we have coming behind that a really excellent consortium comprised of a team at UCSF led by Claire Cleland, partner with Donnelly Therapeutics to try to determine how we can target the DNA itself, which is really challenging from a therapeutic modality perspective, because you need to find some way to deliver the CRISPR-Cas enzymes into the brain across that blood-brain barrier for which they do not normally cross. And so they're working on highly technical methods to develop that and making some advancements there. And finally, complementary to those therapeutic approaches, we recently started to fund a consortium of four wonderful investigators being led by Rosa Rademakers and also Adrienne Isaacs, who both presented their plans and their future work to study a lot of the key elements of C9orf72 repeat expansion biology, both from the perspective of studying modifier genes that may be modifying the penetrance of that repeat expansion and modifying its impact on either motor neurons in the primary motor cortex or motor neurons in the frontal cortex. And they're also studying a lot of the details of the repeat expansions and how they can change across cells throughout the brain and spinal cord in terms of the length and what that means in terms of disease biology. So, and further, they're going to be testing those hypotheses in cell-based and animal models that are really pretty technically advanced and challenging. So they're helping to tee up the next set of therapeutics with providing a better understanding of the specific aspects of the disease pathology.
Thanks, Amy. Yeah, and like with what a lot of we've been talking about, C9 is very complex, ALS is very complex, so it's great to see that we have all of these really incredible groups working on really unraveling those mysteries and figuring out how we can treat C9 ALS among all other forms. And so, Lauren, Chris, I also want to ask you guys what presentation you found the most compelling and what its impact might be forward looking on the field. So, Lauren, how about we start with you?
I think it's hard to say this year. There's a couple that are co-winners. So maybe I'll answer a slightly different question, just building on something that Amy said. One of my favorite things about the meeting that we didn't touch on is that often as these clinical trials are or are not working in real time, there is an amazing willingness of the groups who have sponsored these trials to share and openly discuss the data at the Target ALS meeting. Now, they benefit because you have this great cross section of smart academics, industry folks, investors, everything in between. Um, but I think, building on this C9 piece, I remember that being the first place that I saw the Biogen sense targeting C9 data. And what followed was kind of one of the richest conversations I've ever heard about, could it be the assays? Could it be the sequence? Could it be how the trial was designed and everything in between? So I think I usually come out of the meetings and there's so many important advances, but the discussions that follow and surround them and the questions I think are as important. So sorry, a bit of a cop out.
No, I appreciate that. I think it is like a testament to what we've tried to build at Target ALS with these meetings and just in our ecosystem in general. Like you need to have the voices of all of these different stakeholder groups in order to like get the work done. There's so much, as we've talked about, complex work from the basic biology perspective, the basic scientists to the industry groups and everyone in between. All of these people need to come together because you all have different expertise and can really drive these results faster. And Chris, I will pose this
Yeah.
question to you.
Yeah. So at the expense of not taking favorability, I will say that there were a couple that there were many, many good ones, but two that I really enjoyed. One was more of like sort of a biology reason why it was biology based was one about KCNQ2, which is consortium. I think it's Curalis and Evangelos Kaskinis at Northwestern, where basically when TDP is not working, there's a potassium channel that is made that's dysfunctional. And the really cool thing about this is that whether or not this is causing disease, we don't know, but it's a really good target because there's an incredible phenotype, and they've done a great job at modeling that phenotype and showing that it actually happens. And this is the phenotype in terms of neuronal excitability, and they can model this in human-induced pluripotent stem cells. And it's just a really beautiful story from start to finish. And they're going to try to develop a therapeutic target to correct that. And that to me is just really cool because it takes every aspect of the biology from the TDP dysfunction to an excitability phenotype, which we know happens. The argument whether or not that's happening earlier or later is a whole other question. But that one, and I think one that has really broad implications beyond ALS is going back to the consortia with Claire Kellen's group, where they're developing ways to deliver sort of Cas9 for gene editing of the C9 mutation. And the reason that that's exciting is whether or not it ends up working, this is a really difficult thing to do. There's no better person to do it. But whether or not it ends up working, the idea that they're developing ways to deliver these molecules to the CNS effectively will have implications to, I mean, countless diseases where there's genetic causes or risk factors. And I think that that one is always really just a fascinating talk and consortia, a group to listen to.
Thanks, Chris. And we're winding down on time here. So I want to get to an audience question. And thank you to everybody who have submitted questions. We're going to send out a follow-up email after this, and we'll try to put some additional answers to questions that were asked a lot in there as well as the recording of this webinar. But I'm going to choose one that's a little forward-looking. So now that we're seeing these positive results from tofersen, there's tangible hope that we can treat ALS. There's the data's there. And so looking ahead, what do we think this world where everyone with ALS lives will look like? What will treatment look like? Will it be a single drug? Will it be a combination of drugs? And so we'll do a little bit of a rapid fire. Lauren, let's start with you.
Yeah, I think it depends for folks who have something like an SOD1 mutation, or if we get to a future with a C9 antisense sense. I do think the potential for targeted therapy working well in those populations, doesn't just exist in ALS, it exists in other diseases, and I think there's reason to believe we're on that path. I think for the broader patient population, especially those with idiopathic disease of unknown origin, it's probably going to be combinations, especially as we get into things like neuroinflammation and broad neuronal health modulators. Going after this a couple different ways is likely going to get us some better bang for our buck. But I hope over time we understand more and more of the disease genetically or in a patient stratified manner so we can develop those really tailored therapies, which I think do have generally a bit more horsepower for the patients that they work in.
Thanks, Lauren. Chris?
Um, yeah, I mean, I guess we already do a combination of things, right, for ALS. So I imagine that'll continue. But I think probably different patients are going to require different therapies. Definitely for the genetic causes, but beyond that, I think the biomarker discovery will tell us, you know, maybe one therapy would be better for one set of patients versus another. Um, at the end of the day, we still, so it may look more like, well, I don't want to say too much, but I'm not a clinician. So, but I think, at the end of the day, we still don't know the cause of the disease. Even in the genetic cases, we don't necessarily know why that's causing it. And so we don't know why TDP pathology happens. We know it's bad, but we don't know why it happens. And so I suspect there's probably lots of reasons why it happens. And we're going to be figuring those reasons out. I think it's the next phase. And maybe, hopefully, we'll be able to identify those events that are kind of causing that maybe a little bit earlier. And so now if we're targeting the downstream things, maybe we can target just one step above it and maybe one step above that next. And that's kind of how I think we're going to get there. And so figuring out those two missing pieces, I think, are the next generation of therapies, assuming the downstream therapeutic targets work.
And Amy, close us out here.
Absolutely. I'm going to build on what Chris said just to say, and Lauren too, that there's going to be, I think, a wave of being able to stratify patients to the right therapy better. And that's going to come from these robust clinical data sets and multimodal data sets that Target ALS has been generating along with a number of other groups and that we share out freely. So we can already see that by generating thousands and thousands of clinical and genomic and proteomic data sets from these patients over time that applying tools like AI will be able to break down the highly dimensional data sets and parse out hopefully new biological themes as well as specific drug targets. So I do see better tailored stratified therapies in the future and also drug combinations. I do think and we're a little bit closer to that today than we were before, in that if Curalis is successful with their SAFMan2 ASO and if Trace is successful with their ONC13 ASO, that would be a natural combination to add. And finally, we're funding a company, small biotech called Nucerna, where they're actually developing a single molecule to combine the best properties of antisense oligos with siRNAs to target two different targets simultaneously. So it's coming. Yeah.
Thanks, Amy. And thank you, Chris and Lauren, for joining us today, and all of you out there in the audience. I hope you had an educational afternoon or morning, wherever you are, across the globe. And please stay tuned for that follow-up email with some more information. And I hope you all have a wonderful rest of your day. Thank you.
Thank you, everyone. Bye.
Bye.